You know, every living thing you've ever seen, from the tiniest microbe to the biggest whale, it's all running on the same basic operating system, DNA. But what if we could do more than just read that code? What if we could actually rewrite it?
Well, that's not science fiction anymore. It's one of the biggest frontiers in biology today. And the key to it all isn't some supercomputer or a high-tech laser. Nope. The answer was found right where life began, inside living cells themselves.
That's right. To edit the code of life, you don't use a pen. It turns out nature has already built this incredible toolkit for us, a whole set of microscopic machines, each one designed for a super specific job, cutting, pasting, copying, and tweaking DNA with just unbelievable precision.
So, let's open up this molecular toolkit, shall we? Genetic engineers have this whole suite of specialized proteins called enzymes that they use to manipulate DNA, and they pretty much fall into two main categories based on what they do.
Okay, so on one hand, you've got your composition modifiers. These are the heavy lifters, the tools that actually change the DNA sequence. Think of them as your cutters, pasters, and copiers that rewrite the code itself. Then, on the other hand, you have topology modifiers. Now, these guys don't change the code, but they manage DNA's physical shape. It's kind of like untangling a knotted rope so you can actually work on it. Let's start with the tools that really get in there and change things up. First up, in any toolkit, right, you need something that can cut. And in the world of DNA, those molecular scissors are called nucleases.
So a nuclease has one main job, to slice right through the DNA molecule. It does this by breaking these really strong chemical links, they're called phosphodiester bonds, that form the backbone, the sides of the ladder, of the DNA double helix. This is the most basic, fundamental action you need to do any kind of genetic editing.
But of course, not all cuts are the same. Check this out. Exonucleases, they can only snip away at the very ends of a DNA strand, kind of like trimming a frayed rope. But endonucleases, these are the real game changers. They can cut anywhere in the middle of the rope. And being able to make that precise internal cut, that is what makes modern genetic engineering possible.
So, the absolute heroes of this story, the most important of these endonucleases, are called restriction enzymes. And trust me, they're not just scissors. They are programmable, high-precision scalpels. They are completely fundamental to all of genetic engineering. And just how many of these scalpels do we have in our toolkit? Get this. Scientists have discovered over 4,300 of them, and each one has its own unique target. That is a massive arsenal of tools for editing DNA.
Now, here's what makes them so incredibly powerful. They're actually part of an ancient immune system found in bacteria, designed to find and chop up the DNA of invading viruses. Each enzyme is programmed to find one very specific sequence of DNA letters. That's its restriction site, and it will only cut there. That amazing specificity is like having a find and replace function for the entire genome, letting scientists pull one specific gene out of millions of lines of code.
Okay, so we've got our molecular scissors, but after you cut, you need a way to paste things back together, right? Whether you're rejoining a strand or inserting a whole new piece of DNA, you're going to need some molecular glue.
And that glue is an enzyme called DNA ligase. Its job is the exact opposite of our scissors, the nuclease. While a nuclease breaks those phosphodiester bonds in the DNA backbone, a ligase creates a new one, perfectly sealing the gap and joining two separate fragments into one continuous piece.
And the process itself is just so elegant and efficient. The ligase spots a break, it grabs an energy molecule like ATP for power, and then boom, it forges a new bond, making the DNA strand whole again. It's the essential repairman and construction worker of the cell.
So we can cut and we can paste. But what if you need millions or even billions of copies of a specific piece of DNA? For that, you need the toolkit's copy machine.
So let me introduce you to DNA polymerase, the master replicator. This enzyme latches onto a strand of DNA and uses it like a blueprint to build a brand new, perfectly matching strand, adding one building block at a time. It's the engine of replication, the very process all life uses to copy its own blueprint before dividing.
This amazing enzyme is the engine behind a truly revolutionary technique called PCR, or polymerase chain reaction. See, to make tons of copies, you have to heat the DNA way up to separate its two strands. The problem was, this heat just destroyed most polymerases. The big breakthrough came when scientists found a polymerase in microbes living in boiling hot springs. This heat-resistant enzyme could survive the cycle again and again and again, letting us turn a single DNA molecule into billions of copies. And that, well, that just changed everything.
All right, beyond the big three, cutting, pasting, and copying, the toolkit also has a bunch of specialist enzymes for really fine-tuning the DNA before you get to work.
These next two enzymes give scientists an incredible level of control. Think of them as an on-off switch for the DNA glue. A phosphatase comes in and removes a key chemical group from the end of a DNA strand, which basically tells the DNA ligase, hey, don't paste here. A kinase does the exact opposite. It adds that group back on, giving the ligase the green light to seal the deal. This is all about having ultimate control over what gets joined to what.
Then you have these guys, methyltransferases. They add these tiny little chemical tags called methyl groups onto the DNA. And this can do a couple of things. It can act as an off switch for a gene, or it can be like a shield, protecting that specific section of DNA from being cut by your restriction enzymes. It's just another layer of precise control.
And finally, we've got our topology modifiers, the topolymerases. Imagine trying to unzip a really long, twisted rope. As DNA gets unwound and copied, it gets incredibly tangled and stressed out. These enzymes are the tension managers. They make a quick, temporary cut, let the DNA unwind a bit, and then seal it right back up. Seriously, without them, DNA replication would just grind to a halt.
So, we've seen all the individual tools. Pretty cool, right? But how do they all come together? Let's walk through a classic genetic engineering workflow.
Okay, this is where it all comes together. First, you cut. You use a restriction enzyme, our molecular scissors, to snip out your gene of interest, and you use it to cut open a small circular piece of bacterial DNA called a plasmid. Second, you paste. You take your DNA ligase, the glue, and you stick your gene right into that open plasmid. And finally, you copy. You pop that new hybrid plasmid into bacteria, and they do all the hard work, using their own DNA polymerase to churn out millions and millions of copies for you.
And that's really the central idea right there. Cut, paste, copy. It's this simple yet profoundly powerful combination of natural tools that gives us the ability to read, write, and edit the source code of life itself.
I mean, think about it. From developing brand new medicines to engineering crops that can survive a changing climate, the possibilities are almost limitless. Which leaves us with the ultimate question. Now that we have the tools to engineer life, what do we choose to build?